Hi, my name is Eric Schmidt. I'm on faculty at the University of Pennsylvania, and I've been asked to give an overview of imaging genetics and the role of neurodevelopment in analysis. Um, it's obviously a mouthful, complex field that interfaces several other complex fields, so this is definitely going to be a whirlwind tour. I'm going to have to gloss over sometimes important details, and I apologize for that. I also just want to mention before I get started that a lot of my examples are from our own work, and that's, for the most part, a convenience thing. Just easier for me to talk about it, but there's a lot of other great work out there. So if I entice you into being interested in imaging genetics, please check it out. So I think at this point, probably most people have had exposure to the genetics and imaging genetics, the basic quantitative models that we use, but may not know so much about the imaging part. Almost all genetically informative studies use MRI as the modality, and there are lots of reasons for that. Probably the major reason is it uses non-ionizing radiation, which means it's very safe. It also has very good contrast resolution, which means you can distinguish subtle differences in soft tissue, which is good for the brain. And another reason is that in addition to structure, you can acquire all sorts of other information like functional activation or functional imaging, fMRI, which you probably heard about, tractography or white matter microstructure, perfusion. You may have heard of those things. There's a whole army of other things you may not have heard of yet that you can also get from a MR session. This particular image is probably the workhorse. We've been using it for a couple decades now. This is a T1 NP RAGE, and the way I think about it is it's like a digital downsampled version of the brain. And if you mag it up, which is what I'm trying to show you at the top, you start seeing individual pixels, and each one of those pixels represents a real-world volume, which means like one of the simplest things you could do if you're interested in measuring the brain is to just identify a region and count how many pixels it has in it, and since each one represents a volume, in the real world, you have the volume of the structure. There are other things you can do to it too. You can do all sorts of post-processing. For one thing, each pixel has a different intensity, which means that it gives you some inkling about what's inside it. So you can differentiate gray and white matter. You can also do sorts of other post-processing steps that will give you additional endophenotypes. So when we're talking about imaging genetics as a field, this is what I consider one of the seminal papers. This goes all the way back to the last millennium in 1997, where they were looking at the genetic role on brain sulcations and gyration patterns. And this is about as qualitative as you get. This is a figure of 4 different twin pairs with MRI-based surface renderings of their brain. You can see that within each box, twin pairs tend to have similar patterns relative to if you go across boxes, you see that unrelated individuals have less similar patterns, which was interpreted to be indicative that there was some sort of genetic influence, which turns out to be true. But it's also important noting that they're not perfect. So even monozygote twins don't have exactly the same. So like fingerprints, there is a non-genetic role, just easier to measure fingerprints than to uniquely identify someone based on their gyro patterns, I guess. These days we're more interested in more quantitative measures. This is just to emphasize the fact that at least for structural imaging, it's basically a 3D model of the brain, so you can get specific volumetric measures or other sorts of quantitative measures. In this case, I'm just using a fanciful example of cortical thickness, but you're going to extract that information and generate a family-based record data set, just like you would for pretty much any other family-based analysis, twin-based analysis. So I just wanted to emphasize that and just point out that a lot of imaging genetic phenotypes are nice and that they're normally distributed, and they tend to be quantitative. So that gives us a little more power, which can be useful because even though these data sets tend to be large, they're not as large as like a traditional twin and family study. Nevertheless, we are pretty convinced about some things. So this is just an overview of several different studies looking at large brain volumes, and they differ in sample size, when they were done, but they have one thing in common, which is that if you're looking at large brain volumes, heritabilities are very, very high. Basically, it's pretty clear that genetic factors are dominant, sometimes seem like they're the only thing that influences individual differences in these big things, like intracranial volume or hemispheric volumes or total gray or white matter. We're obviously more interested in regional subtleties because the brain isn't a monolithic structure that just does one thing. It has multiple different functions. And we know that they're regionally localized. So we've looked for other endophenotypes that are a little more specific. Now this is looking at cortical thickness, which is exactly what you think it is. It's how thick the cortex is around the outside. Easy to say, very hard to measure mathematically, but that's not our problem. And what you can see is there's a lot of regional variability, and actually the patterns we see are quite interesting given our understanding of evolution and emergence of certain functions of the brain in humans. So basically we see high frontal heritabilities, higher than other areas, and then perisylvian heritabilities too. And one of the things that was most interesting when we saw it is that the language centers tend to have high heritabilities. So this one back here is the expressive language center. This is ballpark Wernicke's area. This is ballpark Broca's area, the expressive language. I think I said expressive. This is receptive language. And actually this has been replicated in other samples. So this is a landmark study by Paul Thompson, which showed gray matter density, but shows similar patterns. And you even see similar patterns of high heritability in the parts of the motor cortex and sensory cortex that involve the tongue and the mouth. So I put this slide in for a couple different reasons. One is to just show you other examples of univariate heritability measures for different endophenotypes. So this is myelination, arealization, which is surface area, cortical thickness, and cortical folding, which is a measure of gyro complexity. And you can see the patterns are unique between them. There's all sorts of interesting things to talk about that we don't really have time to talk about. But so that's one thing though, just regional variability. And the second thing is just that this is Human Connectome Project data, which is a young adult sample, and it's freely available in public. So I just wanted to put this in here. If you are enticed to look more into imaging genetics, the cost is low. There's an easy access. This one, the ABCD, there are other freely available genetically informative databases that are out there for you. All right, so now I'm going to spend the rest of the time talking about some more complexity. And I apologize that I'm going to kind of oversimplify things a little bit. The reality is that understanding brain structure involves thinking about lots of different things simultaneously. It is inherently a multivariate problem. And we've so far just talked about genetics, but we know that you can't really think about how the brain works without thinking in multivariate space. So first, let's talk about the role between genetics and brain networks. In neuroscience at large, it's becoming increasingly obvious that connectivity relationships between brain regions are very important in understanding brain pathology. The things I think about first are schizophrenia and depression. There's just increasing evidence over time that disruptions in connectivity are important. So we have to understand how to study that. This slide here has nothing to do with genetics. This is just some examples of, at the top, how different ways to analyze multivariate problems in the brain. So one way to look at connectivity is to use a seed region, where you have your favorite region, and you basically measure its relationship to all other brain regions. So at a very fine scale, you can see very fine patterns in relationship with that one seed, and you can actually do that for multiple different seeds. You can do classic statistical analyses like principal components analysis or linear discriminant analysis, if you're familiar with those. And then kind of one of the emergent things that we've been doing more and more is graph-based analysis, which is looking at the network properties of the brain. And that enables you to do a whole new array of analyses where you're looking, even though you're looking at very multivariate data, you can actually derive global measures of network cohesion or all sorts of different things. Some of these will apply to when we're incorporating genetic information too. So that's the top. The bottom here, what I'm trying to show you is just that a lot of patterns start emerging when you start looking at these measurements in multivariate space. So you start seeing, if you look at like structural imaging, you start seeing different areas much more strongly correlated with one another, and they tend to sort. And it turns out that if you look at functional imaging, the similar brain regions tend to sort the same way. And actually, if you look at how things change over time, you also see things that are changing. The patterns are very similar. So you start seeing overlapping correlational patterns in different modalities, MRI-based modalities, which I think is interesting. And I think as behavioral geneticists, we have the opportunity to contribute one more modality, which is the genetic role in making these patterns appear. So this is a pretty crude way of doing that. This is from my dissertation, just looking at bivariate relationships. So you've seen this model before. This is just a bivariate Koleski, and we've basically brute forced looking at all pairwise combinations of relationships in the NIH twin sample. And you can see, well, we have a genetic correlation map, and you can see this is actually now, once we created the genetic correlation matrix, we've clustered it based on a hierarchical cluster analysis, and we see is that the genetic correlations actually replicate lobar anatomy just based on the genetic correlations alone. So there's genetic patterning going on in the brain, even in childhood. And you can actually do other things too. This is a principal components analysis, just a different way of looking at the multivariate space. And this first one here is the dominant factor, and it's frontoparietal. So it's not quite lobar anatomy, but it turns out that this frontoparietal network is well established both in functional imaging, but also in other structural studies that are not genetically informative. So that's just one way you can do it. Here's the seed-based analysis, but this is genetically informative seed-based analysis. So in this case, what we've done is the same thing, model you already know about, a Koleski model, bivariate Koleski. We've picked a seed as one of the two variables, and then the other variable is the 40,000 different points of space and thickness over the entire hemisphere. So mean cortical thickness for one region of the brain versus points in space. And then we've corrected that for all the thousands, tens of thousands of tests we've done. And we see very interesting patterns. Once again, don't have time to go into the great details, but you can see that for the most part, one cool thing is that you see that contralateral homologs, the similar structure on the other side of the brain, actually is very genetically correlated with whatever one you pick, which is cool when you think about it. That's basically what that means, is that like my right-sided superior frontal gyrus actually predicts the thickness of my twins' left-sided superior frontal gyrus, which is pretty awesome, I think. So that's the genetics brain networks relationship. Now let's go to this other one, which is the one I want to talk about more, the genetics development angle. This particular figure or movie is from one of my favorite papers ever by Nitin Gate at NIH, who looked at gray matter density. They looked at it over time. And I think, it's intuitive that the pediatric brain is changing, but to see it in action really influenced how I thought about brain genetics, how things change over time. And you can see a few different things from this graph. One thing is just that it's highly dynamic, right? Like the gray matter density is changing. The age range here, I think, is 5 to 12, 5 to 17 or something like that. So changing pretty rapidly over childhood. The second thing is that it's regionally specific. So the frontal lobe and the temporal lobe are the last to mature, and those actually also turned out to be the last to mature in humans relative to other things. So ontology, replicating phylogeny a little bit. So the next question that follows is, can behavioral genetics contribute anything to understanding these patterns? We've got a kind of an inkling that age was influencing our data with cross-sectional studies. I put this in because I know you guys talked about heterogeneity models, and this is just a heterogeneity model with imaging data. So cross-sectional data set and looking at how age as a moderator influences heritability. So we've got variance on the, so I'm a radiologist, so everything's backwards. So on the left, and then proportional variance or heritability on the right, or proportional variance, not just heritability. And you can actually see dynamic changes on both. And we don't have time to go into the details, but we can do that. Another kind of evidence, indirect evidence that there's change, is looking at cross-sectional heritability at different time points, but once again, not longitudinal. And you can actually see, this is cortical thickness instead of volumes, and you can actually see that you get the sense that heritability is increasing over time, over childhood, and then these two bottom ones are actually older studies. So the Thompson study is adults, just kind of generic adults, on younger side of the life cycle, and VETSA is on the older side of the life cycle, and you can actually see heritability seems to be going up throughout this. So this is from a different group that I just wanted to acknowledge because they've done a lot of great work in longitudinal pediatric imaging. This is the Dutch group, and they have a great sample where they have basically a cohort with 3 separate time points at 9, 12, and 17. This is an analysis they did using basically a traditional Koleski decomposition with some post hoc calculations that allowed them to measure how heritability changes over time. And what you see is that between 9 and 12, you don't see a whole lot. It's kind of static. And then in this kind of, in the puberty range, early puberty, you actually see this big burst of heritability increasing. So you get this sense that there's dynamic effects. And based on their structure, I don't think I have time to talk about it, but structure of their model, they can actually infer whether or not those are novel genetic influences or increases in existing genetic influences. We weren't so lucky to have like nice bins time points. So this is the NIH sample, and it's amazing in a lot of ways. It's huge, at least by early 21st century standards. And it's got a lot of different kinds of data in it. So MZ twins, DZ twins, and singletons are color coded here. And each one of these dots represents an entire brain MRI with 80,000 different measures of cortical thickness in it, or other things. But in this, what I'm going to show you is cortical thickness. You can see some problems with just kind of a generic longitudinal analysis with 3 time points. Like each time point is not equally spaced. So even though we've got a lot of data, it's not easily analyzed. And so we try to think of ways to turn this to our advantage, all this heterogeneity and variability in age to our advantage. And LGC, which you've heard about, is, I think you've heard about it so far, you will if you haven't, is one way of dealing with that. So in this case, we're using age as a dynamic moderator variable that influences the influences of the genetic and non-genetic factors on the phenotypic measure. So it allows us to use all the information, like the exact age of each one at the time of scan. And that allows us to get very kind of fine details of how heritability is changing over the age range. Although we can't say anything about whether or not there's novel influences using our model, but we can get pretty good estimates of at specific time points. And we do indeed see, as we expected, that heritability increases over time. And this is just a model of that. And this is going from 5 to 18. You may have seen this figure before, but this is, like I said, I think that seeing everything dynamically is one of the best ways to think about longitudinal data, and requires movies. So you can see this like wave of heritability change over time, increasing as you get into adulthood, and then with the maps approximating what other groups see in adults. Okay, so we've talked about one of these relationships between 2 of these variables, and then we talked about the other. Now we're going to go for the trifecta. We're going for all 3 at the same time in the last few minutes. This is what we've kind of been working on now. So once again, taking genetics out for just a moment, the neuroscience world at large has been thinking about how different brain regions change over time. And that phenomenon is often called maturational coupling, how 2 different brain regions mature together. And one of our colleagues, Armin Rasmussen at NIH, is one of the pioneers in this. So kind of got the seed in my mind early that we should be looking more at this. And we can do that with making our univariate latent growth curve model bivariate, looking at how 2 different brain regions move simultaneously with one another, which is a much more complicated model, but kind of similar principles. And don't have time to go through the details. Said that like a billion times, but really don't. But now we've got, instead of a static correlation matrix, genetic correlation matrix, now we've got a dynamic genetic correlation matrix, because we know that these things are changing over time. And I'm not showing this to you, but a lot of these relationships are highly significant. But this is actually the correlations. I want to show you how they change over time. You see they start off kind of a little blurry, and then this is going from age from 5 to 18. And you can see that over time, these networks start coalescing with one another at the lobar level. So these are, all these different measures are at the lobar level, which is a little crude, but these models are complicated. So we started with something a little more computationally feasible. Just play that one more time. You can see that basically the relationships within the network, the subnetworks, get stronger, whereas the relationships between the subnetworks gets weaker. Okay. And that kind of makes sense if you look at the literature on spatiotemporal dynamics of gene expression. So smaller samples, but actually having measured gene expression because of cadavers. And you can actually see that these increases over time in the correlations between the regions from fetal to post to adulthood. This isn't the best, actually this isn't the best example, because since this paper came out, they've done better, had data with better spatial resolution. Excuse me, better temporal resolution. So this is what we're working on now. Similar model, but now we're looking at bivariate to what I showed you before with the lobar level cortical, but now we're looking at much smaller regions of cortical thickness, 308 different measures. And you can actually see very fine variations in the connectivity between different brain regions. So we see changes in heritability, which we knew about, but I think what I want to show you most here is you see very fine changes in network dynamics. This is actually, once again, best seen as a movie. These are the lines here. So each point in space corresponds to a brain region, one of the 308 brain regions, and then each one of the lines represents a genetic correlation of 0.8 or above. If we showed all of them, it would be just a mess. And now we're going to play it. No, it's not going to show one. I'll show you one or the other. But you can actually see that these networks are changing over time. And what you see is that similar to at the lobar level, the total number of connections kind of changes and at first decreases, but then certain areas start becoming much stronger related to the entire brain, or at least certain regions. So the strongest is actually in the parietal lobe here. The size of these little balls actually is how many connections they have above the threshold, in this case 0.8. And here's on the other side. So we can actually, once again, see similar things. This is looking from the side of the brain, whereas over on the other one is from the top. And you can see, once again, the strengthening of this parietal connection, especially frontal parietal connection here. So I think this is pretty cool. I like looking at things dynamically. And the last thing I'm going to say, this is definitely longer than 15 minutes, I'm sorry, is, okay, so what? Like, I'm a radiologist, I like anatomy, but, you know, behavior is what really matters. So does any of this matter related to behavioral measures, psychopathology, whatever? The answer is we're still working on it, but yes, we have some evidence of that. So once again, this is going back to before, not talking about genetics. Looking at relationships between IQ and the brain has been kind of a holy grail for a long time. And we found weak associations looking at just raw correlations. And then this paper came out, another landmark paper by Phil Shaw, showed that, that the trajectory, the changes over time in how the cortex develop actually have a pretty strong association with predicting what someone's IQ is. This is interesting because it just basically lets you know that if you're just doing something simple, like looking at a correlation at one time point, you may not actually catch a relatively strong association between IQ and the brain. So we can actually repurpose our bivariate models. Instead of looking at two different brain regions, we look at the brain region over time longitudinally plus IQ. And so this is how the correlations, genetically mediated correlations over time between cortical thickness at many different time points, many different spatial points, and full scale IQ. You can see that it's very dynamic. There's a wave that changes over time, where depending on what time point you look at, you're actually going to see a stronger association. And in reality, probably what we're seeing here is a proxy. Like, it's not like, you know, there's actually a migration of what parts of the brain are most associated with IQ, but more that the maturational change is related, and this is kind of, we're seeing it secondarily. The thing I think this is so cool, and I wish I showed more of this. This is actually just trending out over age, genetic correlations, the phenotypic correlations, that's RP, and this is the proportion of the total phenotypic variance is attributable to genetic factors. Sometimes that's called the bivariate heritability. Well, it's not a great name, but in any case, you can actually see that there's a dynamic change. And the thing I think is really cool is that this plot actually fits very well if you look at the literature, but only if you look at the specific time point. So I'm not showing you all the time points, but this is brain IQ, cortical thickness versus IQ relationships between adults and IQ in adults. So what you see is this strong frontal orbitofrontal involvement, which we see, but only in our 17-year-olds and not in our younger subjects. And actually, if you look at other studies that are in younger subjects that are cross-sectional, you'll actually see that time point is actually more related. So we see genetically mediated changes, and they seem to match up with the phenotypic studies, but only if you look at or consider age as a factor. So that's way over, sorry, but a lot to get through. So just to conclude, I just wanted to give you an overview and show you that that genetic factors are definitely playing a role in explaining individual differences in brain patterning. There's a lot of work to do. We've kind of done like the low-hanging fruit, but there's a lot of more subtle stuff that still needs to be done, especially brain behavioral correlations. We see very different heritability patterns for different phenotypes. And actually, if you read the papers, you can see that there are reasons for that that make sense given the at-large neuroscience literature. And the toolkit that you're learning here actually is very readily applicable to imaging genetic questions. In fact, I think pretty much every model, except maybe the bivariate latent growth curve models, are things that you're learning about in this course. And there are several publicly available genetically informative imaging samples. So if you get the urge, you can probably pretty easily. It's kind of a low threshold for getting involved. And finally, I just want to emphasize, if I haven't already, that the influence of genes on brain structure is highly dynamic, especially if you're doing analyses in kids. You have to think about it, at least when you're interpreting your data. And that's it. I'm over time a little bit, I think, but I think hopefully it was worth it. Thank you for your time. I appreciate it, and enjoy the rest of the course.